Okay, so we've got 90 milliliters of water in this mason jar, and we're gonna add 10 milliliters of dish soap, which is 2 teaspoons. So we're just gonna measure that out like this.
And then the contents of the soap latch on to the fat molecules and remove them from the cell. In doing so, the soap breaks up the membranes that are within the cell, allowing for the cell to burst open and expand the DNA.
Alright, next order of business is adding half a teaspoon of salt to the mixture, 1/4 of a teaspoon of salt. So we're just gonna scoop that out.
And then salt is important because it does not dissolve in alcohol and it binds to the DNA. However, when put in alcohol, the salt solidifies and continues to latch on to the DNA.
Very true. So then we just, what do we do with this? Stir it? Just gonna stir this.
You just cut out the part.
Alright, so we're stirring this mixture up. It's looking pretty consistent consistency, which is great. And that's what I love my hands. And we're just gonna add it to this bag now. Just pour, kiwi smells so bad. That's pretty gross.
Okay, and then you're going to want to remove as much air as possible so you can get a tight seal.
So now we've got our mixture, and we're gonna just add 1 teaspoon of isopropyl alcohol. Yeah.
And it's important that the isopropyl alcohol is iced and that it's not at room temperature because the ice mechanism helps for it to keep the DNA protected prior to extraction.
So unfortunately, this is about as much as we could get into one grasp because there were just some tiny little gobs, not really enough to be seen on video. We're guessing that might have something to do with our salt because we did use granulated salt, but it might not have been finely enough granulated for it to fully dissolve and latch onto the DNA. And we predict that might have been our experimental error, but we're not completely sure. There could have been other factors such as too little soap or other things such as that, so.